A very pleasant morning to one and all gathered in this virtual meeting. It gives me immense pleasure to welcome you all to the second day of this week long journey into the future of our field. After a phenomenal start yesterday, we continue our exploration of the digital and computational frontiers of science. I would like to welcome our respected management, our Secretary Sri Lalit Kumar OJ, our Principal in Charge Dr. Ed Sujata ma'am, our Organizing Convener Dr. G.P. Bhavani, fellow faculty members, and most importantly, our enthusiastic participants joining us from various institutions. It is my distinct honor to introduce our expert speaker for day two, Dr. P. Ravikumar, Senior Scientist and Head at IRB Research Center for Bioinformatics, Erode. Dr. Ravi Kumar is a scholar of exceptional versatility, holding a PhD in bioinformatics, an MSc in bioinformatics, a BSc in biochemistry, and a PG diploma in nanotechnology. This multiple disciplinary foundation allows him to transverse the boundaries of chemistry, biology, and computational technology with ease. His professional journey includes significant role as a research associate at Madras Veterinary College and a project scientist at the Punjab Biotechnology Incubator. Currently, at the helm of IRCB, he leads cutting edge research in computer aided drug design and molecular modeling, phytochemistry, focusing on the isolation and purification of natural compounds, water and soil science, as well as toxicity analysis. With 30 research articles published and experience guiding nearly 80 projects across various academic levels. He is a prolific contributor to the scientific community. His expertise is globally recognized through his roles as a reviewer for high impact journals such as Elsevier Food and Chemistry, Springer's Journal of Food Science and Technology. Beyond the lab, his administrative capabilities are equally impressive. Have managed advanced analytical labs and organized numerous national level seminars and workshops. Sir, we are privileged to have you with us today. Your work at the intersection of bioinformatics and chemistry is exactly what our students need to see to understand where the future of this field is heading. We eagerly look forward to your sessions. The virtual platform is now yours.
Thank you, ma'am.
Thank you, sir. Please proceed, sir.
Okay, ma'am. So, first of all, good morning
Thank you,
to all.
Shobana, ma'am. Yes, sir. Thank you.
First of all, I thank the management and the HOD, Department of Chemistry, um, Jain College in Minjur for giving this wonderful opportunity. So, I am Dr. P. Ravikumar, Senior Scientist and Head the Iyarvi Research Center for Bioinformatics, Erode, Tamil Nadu. Uh, so in this seminar, we are going to see about bioinformatics and in its applications in organic chemistry. Okay. So how we will use bioinformatics in organic chemistry, we are going to see in this seminar. Okay. So introduction to bioinformatics. So what is meant by bioinformatics? It is an interdisciplinary field that develops methods, software, and tools for understanding biological data. Okay, so what is meant by methods, software, and tools? Actually, we know we in our day-to-day life we have seen so many softwares. Right? If you go in grocery shops, restaurant, and movies, you will get the bill, through the software only nowadays, right? So, what is the purpose of the software? To simplify the work. Okay, to reduce the time to simplify the workload. The same way in biology or in chemistry or in other fields, the softwares and tools was used to reduce the workload and the time. Okay, and to do the research in easy way. Okay, so like that in biology, the software means in many software the tools are used nowadays to do research in easy way. Okay, so for that the bioinformatics field was nowadays is using. So what is meant by bioinformatics is combines biology, chemistry, physics, computer science, information engineering, mathematics, and statistics to analyze and interpret the biological data. Okay, so it's not a single field. So if you want to know about bioinformatics, at least you have knowledge in all these basic knowledge in all these fields. Okay, so in bioinformatics mainly includes the computer programming as a part of their methodology. Okay, so in biology, generally bioinformatics uses computer programs for variety of applications. For example, including determining gene and protein function, establishing evolutionary relationship, and predicting three-dimensional shape of proteins. So these are all the functions mainly in biology. Let me see in chemistry in the upcoming slides. So in bioinformatics is the field of computational science, which is useful to analyze the sequence of biological molecules like DNA, RNA, and protein, and is particularly useful in comparing genes and other sequence in protein, other sequence within an organism or between the organism. We can easily analyze using this bioinformatics tools and software. So why I am telling this in biology also, the basic biology, because if you are doing in chemistry, so finally you have to find application, right? So whatever the compound, the organic compound you are finding, so you have to do some applications in biology only. That's why. So for what are the applications you can do in biology, I am explaining here. Okay. So these are all the things, So you're determining gene and protein functions and then predicting predictors. It's all are close related to chemistry. That's why I'm telling here. Okay. So who is the founder of bioinformatics? So Margaret Billy Dayhoff. So she is the founder of bioinformatics. He is an American physical chemist and a pioneer in the field of bioinformatics. Okay. So Dayhoff is known as the founder of bioinformatics. So what she did actually using the mathematics and computational techniques, she did sequencing for proteins and nucleic acids and establishing the first publicly available database for research in the area. Okay. And then G.N. Ramachandran, godfather of Indian bioinformatics, his first major contribution was the discovery of typical structure of collagen. Okay, so collagen is the most abundant protein in the body. It is primary building block of your body, skin, muscles, bones, and tendons, and ligaments, and other connective tissues. So he only found the collagen, 3D structure of collagen in India. That's why he is called as godfather of Indian bioinformatics. Okay. So next, what is the advantage of bioinformatics? Okay, so main advantage, as I told you, the first right, so time consuming. Okay, so in bioinformatics softwares and tools, if you are using in any field like biology, chemistry, anywhere, so why we are using this bioinformatics? Because you already have chemistry, so already biochemistry is there, so many microbiology is there. So why we want to use this bioinformatics? So what is the purpose? Main purpose is time consuming. If instead of using wet lab techniques, if you use bioinformatics softwares, tools, and database, you will get the data or analyze easily in very less time. Okay, then reduce chemical usage. So if you are doing any analysis in chemistry, so you have to use so many chemicals, right? But if you use any this database and tools and software in bioinformatics, then no need any chemicals. We have only the computer and the person who know the techniques. Okay, and then reduce manpower. So we don't want so much of manpower. One person can handle nearly hundreds, 100 compounds. If you want to check the pharmacological activity of 100 synthetic compounds, one person is enough, But if you are doing in the wet lab, so we need so many, at least 4 person we have to allot for doing this analysis and all. So we don't want. So much manpower if you use bioinformatics techniques. Okay, so reduce cost. So it will helpful to reduce the cost also. How? So if you're using chemicals, and if you're doing any studies for checking the your synthesized compounds pharmacological activity, and so what happens? So you have to hire faculty, then hire. We have to purchase chemicals, glasswares. So many things we want. For all these, we need money, right? So, but if you are doing analysis in computer, so no need. You need only the software and the computer. Okay. So the cost will be minimized. Okay. So these are all the main advantages. Then useful in pandemic time. So like pandemic time, like COVID, if it is any research we want to do, so in urgent cases, so we can't know if you are doing any research for finding drug for treating COVID and all, we can't able to do everything in wet lab. So if you are having computer, that means it's a bioinformatics technique, you can easily identify or analyze the data. Okay, so how it will analyze, I will tell you, in the upcoming slides. So how the bioinformatics is useful in pandemic time, mainly in three ways. Okay, so, all over the world in during pandemic time, a scientist try to find medicine in three ways. Okay, so one is repurposing of drug, another one is vaccine design, and another is drug discovery. So what is in the repurposing of drug? So if the drug is already available for some other disease, So what the scientists do, the scientists they took that drug for treating COVID. Okay, so for example, hydroxychloroquine. I think in the COVID time this drug it's highly demanded. Okay, so in all over India, even all over the world, no, so this drug was highly demanded, but this is mainly useful for malarial drug. So this drug is already available. It's completed clinical trial and everything. So in that, why the scientists are using this malarial drug for COVID? Because no, while checking many drugs against COVID, already available drugs. Okay, so if any drug, so because daily in day to day, daily people are so many people are dying all over the world. So we have to prevent the death of the people. So the scientists are trying to repurposing of drugs. So if any drug of other disease, if it is helpful to reduce any single symptom, it will be very helpful, no, to control the death of people. So that's why all over the world scientists took this technique like repurposing of drug. So for this technique, no, suppose they want to check how the hydroxychloroquine or any adriamycin, anything, any cancer drug you can take, antimalarial drug you can take. You want to check in lab, so how many days it will take, right? So it will take no so many days, right? So, but if you use bioinformatics, within 1 hour you will find whether this particular drug no is can able to impact with COVID virus or not. Okay, so that is the advantage. So another one is vaccine design. So like repurposing of drug, vaccine design also one of the technique in bioinformatics. Using this technique only, all over the world, in each and every country found vaccine in within 2 years. So because of that vaccine only, we got that vaccine. Now we are socially mingle, right? So another one is drug discovery. So this is a new drug finding. So far, some all over the world, so many universities and research institutes they try to find a new drug for drug discovery. So for that concept also they used only this bioinformatics techniques, like cheminformatics. That only we are going to see in detail. Okay. So like this, in pandemic time also, the bioinformatics, used in many ways to find drugs to save the people. Okay, so like this, during pandemic time, bioinformatics also very helpful, and these are all the advantages of bioinformatics. Okay. And another one expert about bioinformatics in COVID pandemic time. So in India, so these are the experts. No, they were used the bioinformatics techniques to find medicine or to control for COVID virus. So next one, meaning informatics and databases, online tools and software are available in bioinformatics, which helps to do research in easy way. So that related to chemistry, what are the informatics or databases is available, so that we are going to see in the upcoming slides. Okay. So various informatics. So what are the various informatics is available? So structural bioinformatics, which is tell about structure of biological macromolecules such as protein, RNA, and DNA, and bioimage informatics is a field of bioinformatics and computational biology. It focuses on the computational techniques to analyze bioimages, okay, like cellular and molecular images. And the immunoinformatics, no, it deals with the computational technique and resources used to study the immune functions used in vaccine design. And the wet informatics, which deals with the databases, proteomics, genomics, drug design, phylogenetic analysis, in veterinary sciences, like how we are doing research for a human. The same way, we can use this bioinformatics techniques to find the medicine or find the drug for veterinary animals. In the same way for agriculture also to find the pesticides, insecticides. We will use this bioinformatics techniques in agriculture field also. Okay. Next one is phytoinformatics. So how we will use what is phytoinformatics? It combines the bioinformatics and herbal medicine, focusing on the identification of active compounds in plants for health benefits. Okay. So how we will use the herbal medicines and the bioinformatics to find active potential organic compounds from plant. So this all comes under phytoinformatics. Here the active compounds are secondary metabolites, that is organic compounds. So tox informatics, another one is tox informatics, which is helpful to predicting the toxicity of chemical compounds based on their molecular structure. Then chem informatics, it combines chemistry, computer science, and information science to analyze chemical data and used in drug discovery. Okay, so these are all various informatics in bioinformatics. So now we'll see cheminformatics. Okay, so this one is mainly used by chemistry people. So what is meant by cheminformatics? So cheminformatics is a relatively new field of information technology that focuses on the collection, storage, and analysis of chemical data. Okay, the chemical data includes information on small formulas, structures, properties, spectra, and activities like biological industrial activities. So cheminformatics originally, what is the purpose? Is I told you, in the advantage slide. So cheminformatics originally emerged to help the drug discovery and development process. Okay? However, cheminformatics now plays an important role in many areas like biology, chemistry, and biochemistry. So it is heavily used in drug discovery process. In what are the ways it was used in drug discovery process? In visual screening, molecular modeling, and KSR modeling to predict compound properties, toxicity, and bioactivity. Okay. So what is in the here visual screening? So if you are having a bunch of compounds, so if you want to find the potential lead compound, the potential compound, so you can use this visual screening techniques, then molecular modeling also. So if you are if you want to do a protein and a compound or that means ligand interaction, so we have to build the model of our target protein. So this is the molecular modeling in PSR. So if you wanted to structure activity relationship, so it means so based on the structure of the chemical compound, we will predict the compound properties, toxicity, and bioactivity. Okay, so in cheminformatics, so many tools, the key tools or the following key tools are involved in cheminformatics like molecular representations, databases, and tools and software. So these are all key tools involved in cheminformatics. Okay. So what is meant by molecular representation? So generally, in cheminformatics, molecular representations, smiles of the compound, is very important. So because these smiles and INSI are only useful to find the potential drug. Okay, using several databases and tools. So this smiles only useful as an input for different chemical databases and tools and softwares. Okay, to find the potential drug for any disease. Okay. So what is meant by the molecular representation? So computationally, chemical compounds are represented as smiles and tinges. Okay, so we can't take that compound. So you are making drawing, 2D, 3D chemical compound. You are making drawing, right, in Chemdra or ChemSketch somewhere. Or so we can't take that that benzene ring or aliphatic or ring type structure directly to the computer. Okay, so we have to convert it as smile or inch. Okay, so for these smiles or inch only helpful to find the drug. Okay, so smiles and inch or standardized text-based linear notations used to represent 2D and 3D molecular structures for computational analysis, databases, and AI-driven drug designs. Okay, so generally, and how the smiles will represent, it represent as a human readable flexible strings. Okay, and the ninja is represent as a unique, rigorously canonicalized multi-layered identifier for each molecule. Okay, so how it will look like? So we will see one by one. Okay, so what is meant by SMILES? So simplified molecular input line entry system. So this is called SMILES. So it represents a molecule as a linear text string based on its graph structures, which means atoms and bonds. So not directly from its molecular formula. So how this the SMILES converted into molecular formula, that is one chemical molecular formula or chemical compound molecular formula, how it is converted as smiles. Okay, for example, we take ethanol. Okay, so ethanol becomes how we will mention ethanol in smiles is just like CCO. How the CCO will come? Because the molecular formula of ethanol is C12 at 6O, right, which is CH3 CH2 OH, right. So how will it will become CCO? So let me see, okay, one by one. See, smiles does not start from formula. Okay, it starts from the connectivity, that is Lewis structure. So what is the basic rule to convert SMILE? So atoms are written by element symbol like CON, etc. Okay. So how we will write first? So you have to write atoms in the element. You have to mention in the element symbol. Okay, like CON, etc. So single bonds are implicit. No symbol needed. Okay. If any single bond is there, you don't consider it. You just ignore it. Okay. So then hydrogens attached to carbon are usually omitted. Okay, so while converting SMILES from chemical formula to uh, SMILES, canonical SMILES, so we have to remove hydrogen attached. Hydrogens attached to carbon are usually omitted. Then you write atoms in connected order. Okay, so like this, the SMILES, this came. How? See, I will explain you. This is the Lewis structure of ethanol, right? So here, what is the condition? So we have to write element in CON. So CON. So in this, whatever the C and O is there, you will write C C. You see this C6, C12, H6O. So here you will write only, you take only this C. So what is the rule? Single bond are implicit. So we don't take no bond between this. So you don't take this bond. So if you remove this bond, what will have? What will be here only? So if you remove this bond, then hydrogen attached to carbon. So you have to remove all these hydrogen, right? So all are attached to carbon, right? So you will remove. Then you will remove this. Uh, you will remove this single bond. Okay. Then so hydrogen attached to carbon is usually omitted. So what will be remaining? C, C, and O. See, the smiles for ethanol is CCO. So you write atoms in connected order. CCO. So now, so like this only, this molecular formula of ethanol is converted into smile, like CCO. Okay, is it clear? And next one, so INCI. So what is in the INCI? International. This one is another representation. Okay, the two form of in the two while doing chemical analysis using computer, I will represent the chemical compound in two form. One is smiles and another one is INCI. Okay, so just now I explained the smiles how we will convert the chemical formula into smiles. Just now I explained. So next one is INCI. Okay, so how the INCI. So what is this smiles and the INCI? These are all useful in so these are all so these are all two forms. Okay, so which is used for computational analysis. So the first one is just now I explained this smiles and now I will explain this INCI. Okay. So international, what is in the INCI? The International Chemical Identifier. So it is a standardized layered textual identifier developed by International Union of Pure and Applied Chemistry (IUPAC). General structure of INCI. So uh, how it will look like? So INCI string has a layer separated by backslash. So what is the formula to convert a chemical formula into this INCI style of representations? So this one. So INCI equal to version backslash formula backslash connectivity hydrogen charge and CD like this. Okay, we have to mention like this. Okay, from formula chemical formula to INCI format. Okay, so how it will convert for the same ethanol? See, this is the example for ethanol. So this is the INCI representations of ethanol, and this one is CCO is smiles. Okay, so how we will convert this INCI oneness? See, we will first divide it into four category. So four division, right? INCI oneness is one. Then this is on C2 H6. So the molecular formula is second one. and this one connectivity is third, and the hydrogen attachment is fourth. Okay, first one is um, this one, the Inchi oneness. What is in the Inchi oneness? So Inchi version uh, 1 with what it is, what this 1 indicates the Inchi version 1, and S indicates the standard Inchi. Okay, so Inchi equal to oneness, which means standard Inchi version 1. Okay, so the how we will write this one. This is just like molecular formula of the compound C2H6O. So here, what it means, I think chemistry people, you know this already. So it means 2 carbons, 6 hydrogens, 1 oxygen, right? So next one is C123, the connectivity layer. So this depends how heavy atoms, that means non-hydrogen atoms are connected. So atoms are numbered internally in canonical order. So for ethanol, so numbering becomes, this is the formula, right? CH3, CH2, O. So we will consider this first CH3 as first atom, second one is second CH2 is second atom, and O is the third atom. Okay, and here the connectivity is 1 to 3. Okay, so meaning what it means, so atom 1 is bounded to atom 2. Okay, this one, if you write no CH3 CH2O, see, the atom 1 is bounded to atom 2, atom 2 is bounded to atom 3, like this. So how it is connected with the carbon? See, 1, 2, 3. So carbon is connected with 1, 2, 3 like this. Okay, which means this one, C1, 2, 3. Next one is how we will write this one, hydrogen. So this is the hydrogen part, right? Hydrogen layer. So this defines where hydrogens are attached. The format is, how we will mention, atom number H count. So how it will write from reverse. Okay, so this is 1H3 and 2H2, 3H. What it means? Atom 1 has 3 hydrogens. Okay, here atom 1 has 3 hydrogens. Atom 2 has 2 hydrogen, means 2H2. Then atom 3 has 1 hydrogen, which means 4H. See, CH3, CH2, 4H. Okay, so finally the distribution is CH3, CH2, OH. So it it's written here reverse. Okay, so like this, the inch will converted from molecular formula of any molecule. So after converting this only, we will use the data for input for many chemical tools. Okay, so this you no need to you no need to convert anything while doing research or analysis. The database will already have the data. The converted data will already have in the database. Okay. See how the engine is actually generated, the algorithm. So what is the algorithm? First to read the structural input. So I told so you no need to do anything for convert any molecular formula into smiles format or NG format. And the software and the database will, I mean, if it is available in the software database, it's already the NG and smile data will available. Or if you find any new synthesizer compound, if you use any ChemSketch or ChemDraw, if you use any tool, that tool will automatically convert that molecular formula or structure into uh, canonical smiles or NG, okay, NG format. So how it will convert? So the algorithm, the basic algorithm for conversion of the chemical structure into smiles or NG format. Okay, so read first. It will read structural input from mole file, then remove implicit hydrogens, then canonically number atoms. So generate layers. So formula, I told formula, connectivity, hydrogen position, and if it is any charge chemistry is there, then isotope is there, it will attach. Okay. So like this, the algorithm will be taken by the tools to find smiles or INC format. Okay. So INC is not a handwritten. No need to do any handwritten. It is computed by software using a VPC INC algorithm. Okay. The smiles and NG is used to, so where we will use this one to represent 3D, 2D and 3D molecular structures and use it as a input for many chemical databases And the tools for computational analysis of the chemical compound, like the information on small molecule formulas, structures, properties, spectra, and the activities like the biological activity or industrial applications and the toxicity. You want to find all these things using computer. You should know this smiles. Okay, so with the smile data. Without the smile data, you can't able to find all these things using computer. If you want to use your chemical data in the computer will take only these format, this files format. It will not take directly the compound. Okay, it will convert it as if you draw the compound in 2D form, it will convert it into smiles. Then that smiles only the computer will take for analyzing and predicting these results like molecular formula, structures, properties, spectra, activities, and all. Okay. So like this, what is the purpose of this all? So it's helpful mainly to helpful for the drug discovery. Okay, drug discovery and development process. We are using this informatics and their key tools like science and NGs, database tools and all. Okay. So what are the major chemical databases are available, um, which is helpful in drug discovery process in the chemical informatics field? So NCBA, okay, this is the authentic database all over the world. People are using for finding the chemical compound information, NCBA PubChem, then ChemSpider, CHEMBL, Impact, DrugBank, and HMDB. So this one is PubChem, ChemSpider, and CHEMBL. You will get chemical compound informations like so these informations like molecular formula, structure, properties, spectra, any biological activities, toxicity, and synonyms, other names. Okay, so who is the vendor? So all the details you will get it in this database. Then impact, this is mainly the medicinal plant phytochemical database. If you are working in phytochemistry, you can know for your comparison purpose you can use this database to get the phytochemical data. Okay, so drug banks, so it's a drug database. So in this database, you will get so many drugs. So currently available drugs, details you will get in this database. So what are the drugs are there? So what is the mechanism of the drug? So how many phases it completed like clinical trials? So the phase one, phase two completed, phase three completed. Okay. Then what are the disease? This drug will useful. So what is the symptoms will come if you use this disease, or any side effects will come or not. So all the details available, for any drug, any approved drug. Okay, any approved currently using from any disease, right? So that drug information you want for any if you are doing any comparison study. Suppose if you are doing any anti-cancer study, if you want to compare with the currently available drug, you want to check the mechanism of action of currently using drug for comparative purpose. You can use this drug bank database. You can easily find the currently available drug uh, full details. Okay. Then HMDB is a human metabolome database. So what are the metabolite available in human body? If you small molecule available in the body, so you will get all the details in this HMDB database. Okay. And this is the homepage of NCBI, and this is the PubChem. In NCBI PubChem, you will get this database, PubChem database in NCBI. And this is the homepage of ChemSpider, and this is the homepage of CHEMBL, homepage of I told Impact, and this is the drug bank database, the homepage of drug bank database. This is the homepage of HMDB. So next tools. So what are the tools are involved in the chem informatics? What are the tools we can use? So like first one is ChemSketch. So the free tool only. The students you can use to draw your chemical compound for using for analyzing computer. You can use this ChemSketch tool. So chemical compound drawing tools. And next one is work on graphic file format conversion tool. So suppose if any software is asking one file like MOL file, SDF file, or PDB file, you can convert using this open source software. You can convert one file format to another file format. These all are free only. All I am telling here, all are free software. You can use it. If you are a student or scholar or faculty, you can use this tool for your research and other academic purpose. Okay. And then Switzerland is also a toxicity prediction tool. It is also free tool you can access in your laptop itself. So if you are finding any compound, organic compound, so if it is giving any side effects or not, so after synthesizing it, if you have a 2D structure, you will convert using this chem sketch. You can draw it and then convert it as a smile using this chem sketch or chemdra. If you know chemdra, you can use chemdra also, but that is commercial one nowadays. Then using this tool, you can convert the chemical structure into smiles. Then you will use the smiles in Physiognomy. Then you will find toxicity of the compound you synthesized in your lab. Okay. So the next one is Protax 3.0. This is also toxicity prediction tool. So some parameters if not available in this Physiognomy, you will get some other parameters, additional features or related to toxicity, you will get in this Protax 3.0. Okay, so these are all the toxicity prediction tool. You can use it for finding toxicity prediction and in Bioinformatics mainly for the drug discovery purpose. Okay, and this is the home page of that game stage. It's open Babel. This is a Switzerland home page. This one is Protax 3.0 home page. Then softwares. So softwares in Bioinformatics. So what are the softwares we can use mainly for drug development? So if you want to visualize your compound, you can use this Pymol, Resmol, and Jymol, and Discovery Studio Visualizer also you can use. Then Pyrex, Autodoc, Autodoc, these are docking software. That is molecular docking software. You can use it as a free software. You can use for doing um, for finding protein ligan interactions. Okay, that means protein chemical compound interactions. You can use this one. Then series chemistry. As I told you, this is a free tool helpful to draw the structure of chemical compound. The same way, the ChemDraw it's a commercial software to draw the structure of chemical compound. Then Discovery Studio, Glide, Soft Gold, Flux Actis are all molecular docking software. These are all used in mainly drug development process. Okay. And in our institute, we are using Spirex and Discovery Studio for finding drug and the synthetic organic compound and protein interactions. So if anyone is interested to your molecular docking studies, you can contact us. So we'll help you to do this molecular docking studies and the informatics approach. Okay. So I will give you some demo for how to use this canonical smiles, how to get the canonical smiles from chemical database, how we will apply in tools. Okay, so I will give you any one tool. Sir, is it possible to share the screen? Okay, I will. I'll present another one slide, then I will. Sir, how can I present this slide, sir? Hello? 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 Sir, is it over program? Sir, I can't able to share that slide, sir. Okay, I will use share the screen. Okay, I will tell you. So how to get data, the smiles from database, then how we will use in the tool. Okay, so just one demo. So this is one. Sir, my voice is audible, no? Okay. So you have to give any name, any chemical compound name like this. Then take a smiles of this compound. See here in database, you will get all the details like the database is this particular compound ID, then 2D and 3D structures, the safety measures, molecular formulas, synonyms. So many things we discussed. No? So many things is available in the chemical database of the particular organic compounds. So from here you will get the canonical smiles. Okay, so you copy the smiles for this anti-cancer drug doxorubicin. Then using this SwissAdMe, this is a toxicity prediction tool. Click run. So you will find the result like this. Okay. So this indicates how your synthesized compounds or drug is toxic or not. See, so you will see here the Lipinski rule. If it is Lipinski rule, no, obey the Lipinski rule. You can use this drug. Okay, this one is synthetic compound. So it has side of this anti-cancer drug. If you take this one, it will give so many side effects. That's why the Lipinski rule is not obeyed. You check the Google. So what is in the Lipinski rule? And so if you are synthesizing any compound, any organic compound, with the compound should obey the Lipinski rule of 5. So first you will check the Lipinski rule of 5 here. So after that you will check the 6 parameters like lipophilicity, size, molecular size and polarity. So insolubility, saturation, flexibility. So you have to check all these parameters. So how here if you move the cursor you will get the details, the notes. So lipophilicity should be minus 0.72 plus 5, then size of the molecular weight of the molecule, which is 150 to 500, then polarity should be 20 to 130. So insolubility should be minus 6 to 0. So all the this is the standard value. You for your compound you will get the value from this tool. Okay. See, lipophilicity means x log T3 value. See the x log T3 value you will get 1.27. Okay, then another value, the size, molecular weight means which should be 150 to 500. So what is the molecular weight? It's above 500. So it's toxic because it's synthesized compound, synthetic compound. It's a toxic, but nowadays for cancer patient they are using this drug only. So like this we will get. So how it is, gastrointestinal absorption is low, then blood-brain barrier permeation is low, no. Okay, so like that you will identify the pharmacokinetic property here, lipophilic nature here, and physicochemical properties of the drug you will find here, the water solubility you will find here, drug likeness you will find here. Okay, if you click this one, the so boil deck, so it will so whether it is uh, toxic or not, you will easily find. See, it is not in the, it should be within this. So if the molecule is highly absorbable, it cross blood-brain barrier, the molecule should be here. See, here is no molecule because this is toxic. Okay. But if you choose another one, you will find compound. If you choose any natural compound like quercetin, you will get. If it is quercetin, if you take, you will give good result. That doxorubicin is a synthetic compound. That's why it gave side effects. See, in boiled egg, you will get. See, its absorption is highly absorbed. It will come like this. Okay, so like this, you will predict the toxicity. If you want to learn more, so you can attend our upcoming workshops in our center. So you will learn how to use this data for your research purpose. You will learn. Okay, so that's all about demo. So I will tell you. I will just explain about our center. So what we are doing, it will be useful to you. You can contact us if you want any research in the area of research service in the area of chem informatics. Okay. So this is our center about IRCB, Iyarvi Research Center for Bioinformatics. So I am Dr. Ravikumar, Senior Scientist and Head of this institute. And this is our contact number and email ID, website. So we are doing as a hub for yes, hub for institute research. It was established in Europe, Tamil Nadu, India. The objective of the center is to provide research training to the students, research scholars, faculties, and scientists to carry out applied research in the area of life sciences, food sciences, nutrition, organic products and toxicity analysis, agricultural, veterinary sciences, medicinal plant, natural products, nutraceuticals, phytochemistry, Siddha, Ayurveda, and traditional medicines, medical, dental sciences, pharmacology, pharmaceutical sciences, organic and analytical chemistry, and computer science. These are all the fields we are working. So I have worked in all these fields in different designations in many institutes. So that's why we are doing in all these areas. Then we are collaborated with several research institutes and colleges in both abroad and India. And our aim is to provide research training, quality research support and services in global standards to the students, research scholars and faculties and scientists from academic and research institutes and companies also to carry out applied research in the center individually. So this is our vision and mission. This is our research area. Okay. So computer drug designing, macrodynamics, machine learning, drug discovery, proteomics, genomics, evolution biology, food science and technology, organic products and toxicity analysis, medicinal plants, mainly natural products, nutraceuticals, and phytochemistry, and botany, zoology, biochemistry. So these are all the fields we are working. Okay. And so the service we are providing to the students and research service and data analysis and collaborative research and publication support services and scientific report and content writing and government and private projects. So individual research and product development and contract laboratory services and business and research consultant services, value-added courses we are conducting for students and certificate courses and faculty development program. So these are all the services we are providing in our center. And trainings, we have trainings we are giving to students of 5-year project, summer internship, winter internship, studentship program, and skill development program, seminars and conference, seminar cum workshops, hands-on training, and some quiz program we are conducting. Then both online and offline mode we are conducting all these trainings in our center. Okay? And IRCB welcomes collaborations from the faculties and MOU from institutions, and these are our various publications, right? We are working in mainly natural products, so we are doing so many things. So this is a veterinary field, and we are working in all, not only in natural products. We are main focus in bioinformatics and natural products. The main focus, but we are working in different areas. So it's for human, and this is for veterinary, okay, veterinary animals, and this is for a human. Okay, this is also for human. So we are working for in several areas. Okay? These are all our applications. So this is our contact number. This is our social media page. If you are in any of the social media, you can follow us for the upcoming updates. Okay. So, any doubts?
Hello, sir. Uh,
Yes.
Thank you, sir. That was a very insightful session from you. We have few questions from the
Okay.
students. I'll post it on the chat now,
Okay.
sir. This is a question from the first student.
Okay.
Let me see here. Yes, sir. Did you get the question, sir? On the chat.
Chat box lo varlinge.
Okay. Sir, I'll read out the question, sir.
Okay. Okay. So we often hear the terms stemming from. Yes. Yes. I got. I got.
Okay.
Together in simple terms, how does these
Two
two
terms differ. Two terms differ by the meaning.
nothing, ma'am. Okay. In chem informatics, mainly deals with the chemical compound. Okay, chemical compound and chemical databases and tools like that. And bioinformatics mainly deals with the gene and protein like that. In biological applications, no, the bioinformatics will do. And chem informatics will mainly work on chemistry areas, so like chemical formula, chemical databases, tools. I told you know in the seminar. So then drug discovery process, both in bioinformatics and cheminformatics are both are involved, but in chemistry angle, without no biology in that in cheminformatics. But in bioinformatics, both chemistry and
Chemistry.
biology involved. So that is the difference. Okay.
Okay. Okay. Okay. So this is a question from one more student.
Okay.
Okay. So you mentioned that we can design drugs on a computer. Does this
Hmm.
mean we can predict if a medicine will be effective before we even step into the laboratory to synthesize it?
Yes, ma'am. So we can, if you use computer softwares, before going to the laboratory, you can find whether it is suitable or not for any particular disease. For example, if you synthesize, 10 compounds, 10 organic compounds, so how we will know whether it is which compound is suitable for any particular drug? How we will know? So if by wet lab only you have to till now you are analyzing, right? But it will take so much of time. But if you know this bioinformatics like cheminformatics techniques, you can easily identify with the computer. So whether it is having potential effect against any particular disease or not, you can easily identify. Okay. So for that purpose only, we'll use this cheminformatics. Yeah.
Thank you, sir. This is another question from another student.
Okay.
So, as a chemistry student, besides from laboratory skills, what are the basic computer softwares and programming languages they have to learn for to enter into the field of drug delivery?
Uh, if you want to become expert in cheminformatics, so you should know. I told you know the database. What are the database? I told you. First, you should know about the input form, that is smiles and INC form. So what is that? You have to aware of this one. Then you have to use databases. Then I told you know these databases. So many databases is there, but what are the database I told you? In the seminar. So that is most familiar one. You can use for your research purpose. Then tools. So what are the tools I told you? You can use it. So all are only free only. You can use it in your laptop or computer. Then the software. So for docking purpose, so many docking software is there. Commercial and free software is available. You have to learn for drug discovery process. You have to learn first the database, how to use what are the databases are there, how to use the database to take the data, then how to use the tools to find the toxicity and all. Then for drug discovery process, there are several processes there in drug discovery. So if you learn the tools which I told you in the seminar, that is more than enough to find the drug development. Then softwares for drug discovery. In our center also we are providing the Pyrex and discovery studio we are doing. So if you want to learn in detail, in hands-on training in online or offline, you will contact us. We will help you for doing the drug development and delivery. if you are a scholar or a PG students, you will learn all these things. Okay, how we will use computer for drug discovery.
Thank you so much, sir. So I'm handing
Thank you.
over the session to Miss Ashwini for vote of thanks.
Okay.
Good afternoon to everyone joined us on the online international seminar. It is my honor to give the vote of thanks for the second day of this week long international seminar organized by the Department of Physics, Sri Center College, Nadu. First of all, we sincerely thank our respected expert speaker, Dr. P. Ravikumar, senior scientist and head of Iyarvi Research Center for Bioinformatics. Sir, thank you for sharing your valuable knowledge, molecular modeling, bioinformatics, and for giving us a wonderful trust to this seminar. Your session, Advantages of Bioinformatics, Collagens, Molecular Representation, our students and faculty members understand modern developments in bioinformatics. We express our heartfelt thanks to our manager and secretary RAVIKUMAR OJ and our principal Dr. N. Sujata for their constant support and encouragement. Our special thanks to our organizing convener and head of the department, Dr. G.P. Bhavani, for organizing this meaningful program and bringing expert knowledge to young minds. We also thank our students and faculty members for attending and actively participating. We hope today's session inspired you and you will help you throughout this week. We look forward to meeting you all tomorrow at 11 a.m. for the next session with Dr. N. Karnamurthy sir. Once again, thank you everyone. Have a pleasant afternoon. Thank you.
Thank you.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.